Welcome to the lecture series on vitamins and nutrition. So, in today's session, we are going to continue from where we left in the previous session. We were discussing on vitamin A. So, if you look at the aspects of vitamin A in the previous session, I explained you about the transportation of vitamin A. We spoke on retinol binding protein, and this retinol binding protein, I want to add little bit more on retinol binding protein. And this particular protein has lots of significance across species. Although we are discussing about these vitamins exclusively in terms of deficiency diseases and metabolic functions in higher organisms, in our case we are dealing with human system, but you find retinol binding protein and similar such carrier proteins being present across species. You find even retinol binding protein present in nematodes. That might really surprise you. Then you might wonder whether other organisms also need vitamin A. Yes, it is true. Wherever you find retinol binding protein and its close association with vitamin A gives a suggestion or a clue on its role of the way in which vitamin could be utilized. So, vitamin A being primarily utilized as an essential vitamin for metabolic functions in human beings also finds its place in utilization by variety of living species including nematodes. In fact, there is a study which goes and says that retinol binding protein could even be used as a immunomodulatory substances, and that can have a direct impact on the way vitamin transportation happens in nematodes. Now, we are going to look further on the transportation of vitamin A. So, as I told you earlier, the vitamin A being transported from the intestines goes to the liver and being carried by retinol binding domain and goes to different parts of the tissues. So, here we have a summary, and we look at the role of retinol, retinal, which is the aldehyde form, and the retinoic acid. So, you see over there the storage of vitamin, and as I told you earlier, the retinoic acid is being stored as a retinol palmitate. And look over here. So, under normal conditions of storage, the reserve of vitamin A from a well fed person with a balanced diet could go up to 6 months to sometimes even 9 months or more. That is really good. So, you mean to say if a person would have to deplete the sources of vitamin A, it would take a minimum of 6 months for the person to completely utilize all the sources of vitamin A. So, means what? We somehow manage to have a storage part in our body, and within this time interval of 6 months, the person could very well have an exposure to vitamin A rich foods, and he could always restore back the reserves. But in deficiency states, that does not happen, and that is one of the primary reason in malnourished environment where children or adults who have been constantly deprived. So, that could happen in situations like famine, where access to quality food would be a problem, or maybe it could happen in people who are on an expedition or on an adventurous journey. So, these are all artificial conditions, but famine and constant malnourishment could also be a source of vitamin A depletion in our body. So, they are not like readily excreted. So, you see over there the vitamin A is broken down into the liver, and the components are being eliminated, being a fat soluble components. So, you see over there we are going to look into the metabolic functions of vitamin A. So, vitamin A is basically utilized in these 4 aspects of health. First aspect being the vision, the second being the growth, the third being the reproduction related functions, and the fourth one being maintenance of epithelial cells. I have already hinted on how vitamin A might play a role in all these 4 metabolic functions. Now, I am going to elaborate more on the biochemical aspects of how vitamin A could play a role in vision. So, let us start from the role of vitamin being used by eyes. So, this one is what we call it like the Wahl's visual cycle, is a very important pathway in which the retina of the eye is able to properly process or convert light energy into electrical impulse. So, in other words, the photosensitive pigment is fundamentally a pigment which is manufactured or synthesized in our body by vitamin A. So, you look over here, a process by which light falling on the retina and the conversion of this light energy into chemical energy and eventually being converted as an electrical impulse as a direct consequence on the way in which vitamin A is available for the body. Suppose if we do not have sufficient amount of vitamin A, directly that will have an impact on retinal processing of information and directly that will have an impact on vision. So, you see over there, there are two kinds of cells in the eyes, especially in the photosensitive area of the retina. So, you would have studied about this probably in your lower classes, and you know them to be as rods and cones. So, rods and cones. So, rod cells are responsible for black and white image. In other words, if you have to look into the details, the enrichment of details, rod cells will take care. And cone cells, what we call them as, depending on the morphological appearance under microscope, how they look like, they take into consideration the color aspects of the eye. So, rod cells and cone cells both are responsible for the vision that we have, the way we appreciate the surrounding of our environment. So, you see over there, if you look into, if you want to look at the position of retina, so you see over there at the back of it, this is the retinal screen. Very interesting enough, although we are going to look at right now the role of vitamin A in the processing of what you call it as the visual purple, but you will see later, the later aspects of the slides, that vitamin A is also playing an essential role in which the light could transmit across. So, for healthy cornea, you need vitamin A. If you do not have vitamin A, they will directly affect the way in which the corneal tissues, the epithelial tissues are formed. If there is a damage in the epithelial tissues, the cornea can be affected, can get keratinized, and it will even affect the way in which light could refract across. So, you need vitamin A for a healthy cornea, you need vitamin A for the overall epithelial components of the eye, you need vitamin A for the active retinal visual purple screen where the information is going to be processed. So, for this entire eye, you need a proper supplement of vitamin A being regularly got through our diet. So, now we are going to see. So, you look at these pictures. So, the light falls on over here and you see over there the ganglion cells, and then you see over there the bipolar cells, and then this is the overall picture where the light falls from the retina directed exactly on the retinal screen, and there above this retinal screen or the retinal screen underlying tissues, you find the ganglionic cells and the bipolar cells. So, the rods and cones are basically the photosynthetic layer where you have the photosensitive layer being nourished by a photosynthetic pigment. So, you see over there, it is quite interesting. In a normal vision, okay, it depends on retina. Any mechanical damage to retina can lead to loss of vision, but the one which I am talking right now, I am talking about the deficiency based retinal damage, the integrity of retinal cells. So, you see over there in the retina, the vitamin A in the form of retinal binds to a protein called opsin. So, opsin is a protein, okay, and rhodopsin is actually a combination of opsin with retinal. So, you see over there 11 cis retinal in conjugation with opsin gives you rhodopsin. So, rhodopsin is called visual purple. Okay. So, we have one more thing called visual violet, which is also a photosynthetic pigment being utilized by cones. Now, our attention let it be on rhodopsin. Although retinal is required in both the cases, rhodopsin is basically the photosynthetic pigment in the retinal cells of rod category of cells. So, you see over there, this is called the Wahl's visual cycle, and we attribute the contribution of Dr. George Wahl, and he did an excellent study in elucidating the way vitamin A is required by us in processing the required visual pigment present in the retina. So, you see over there, you find, as you could see here, 4 different layers. So, the photoreceptor, then the retinal epithelium, then the blood in which these metabolites are being processed back and forth, then you have the liver. So, liver being the storage organ or the reservoir where vitamin A is available, and this basically goes via blood through retinal epithelium and finally reaches the photoreceptor. One of the reasons why the retinal layer is highly profused with capillary cells, capillary blood vessels, makes them a good source by which instantly the blood could reach the surface of retinal layer, the photosynthetic layer. So, from the liver you find there the active form of retinal, which is the cis form, goes. So, the cis form basically you see over there goes over there, and the cis retinal gets converted into 11 cis retinal, and there it combines with opsin to form the photosynthetic pigment visual purple. And when light is exposed on the visual purple, it gets bleached. Immediately there would be photoisomerization reactions happening, and it's been broken down to give all trans retinal. Trans retinal is basically, I would say, the metabolite resulting from degradation or making the retinal inactive once the job is done. You see there one more enzyme, the retinal isomerase, to some extent converts some amount of trans retinal to cis retinal, but eventually this trans retinal would go into systemic circulation and goes to the liver, and there you find it's been converted. Now, in this case it is a reduction reaction, it gets converted to trans retinol. So, it is an aldehyde compound which gets converted into an trans alcohol, trans retinol. This in turn gets converted by an isomerase to cis retinol, and eventually the cis retinol will find its way to high through blood and through the retinal epithelium. So, this is Wahl's cycle. So, this keeps happening constantly. See, even as I am talking to you, even as we are looking at the screen, and even as we are trying to concentrate, you have to remember this Wahl's cycle is happening in our eye. So, and you are able to visualize how quickly things are being replenished and made available. See, you could always imagine we have such a sensitive light sensitive pigment in our eye which is being constantly replaced. So, imagine in olden days the camera where you have to load the film, and the film would basically be active silver halide coated template, and the moment it is been exposed to light, there would be bleaching. So, probably in the digital age the same thing is being taken over by photosensitive layering of cells, and these photosynthetic layering of probably the silicon chip present over there and the sensors, what we call it, basically do the job. This is being constantly replenished. But if you consider a chemical environment like high, what we would see as fine details are being constantly recorded. So, you like literally a flash play the image and the image is immediately processed again. So, everything constantly keeps happening, and we move from a region of a very bright environment to a dark environment, and our eye basically does all this. Remember, this amount of retinal required for the job, the cis-retinal, is synthesized during the dark period or when your eyes are being exposed to darkness. So, that is a time interval in which your eyes would replenish the source of cis-retinal. So, that is one of the reason when you walk from a bright environment and suddenly you go into a dark room, you find it takes little time for you to adapt yourself to the dark environment. Normally it does not take forever, it takes probably some minutes, maybe I would say minutes or also more when you are really healthy. So, within seconds you start adapting. So, this is what we call it like dark adaptation, and vitamin A is extremely essential. In fact, deficiency in vitamin A will have a direct impact in which we would adapt ourselves from a bright environment to a dark environment. In fact, they use this as a diagnostic criterion and they call it like dark adaptation feature or a dark adaptation test. So, you see over there, this is the picture, the light falls on and then you have the all cis retinal isomerizes to all trans retinal in the rhodopsin, and all trans retinal separates from the opsin, and then it goes over there and the opsin triggers a cascade message to the brain, okay, and this is how the cycle happens. So, this is what we mean by the visual cycle. Okay. So, now remember, cis retinal is the active form of retinal and all trans retinal is the inactive form of retinal. So, metabolism of cis to trans would happen whenever the rhodopsin pigment gets exposed to light. So, you look over there. So, vitamin A isomerizes from its bent cis form to the straight trans form and detaches from the opsin. So, there is lots of stereochemical changes happening in the structure. So, retinal released in this process, it is converted into trans retinal and then to cis retinal to begin the other cycle. So, this keeps happening on and on. So, you see there, as I told you about dark adaptation, what we also call it as photo bleaching. The bright light depletes rhodopsin, completely wipes off rhodopsin, but the amount of rhodopsin lost and the intensity in which the bleaching happens is interpreted as an electrical signal through the optic nerve, and this information is carried further to your brain, and in the visual area the information is processed. So, it is so quick, within minutes rhodopsin is replenished. So, we do not continue to be in darkness, but vitamin A deficiency will definitely have an impact in dark adaptation time. So, you see over there how the bleaching happens when exposed to the light. The color of the rhodopsin changes from, I would say, red to yellow. So, you do have certain intermediary molecules. Okay, you see there rhodopsin getting converted into pre-Lumi rhodopsin, and this pre-Lumi rhodopsin gets converted into Lumi rhodopsin. Then you have metarhodopsin, and then you have the metarhodopsin 2, and finally the all trans retinal separates from the opsin. So, this is the pathway in which rhodopsin would fundamentally being degraded to its components all trans retinal and opsin. So, this is how it happens. So, every stage you find a metabolite being formed. So, this way how rhodopsin gets broken down and forms the altrans, retinal and opsin would tell you the time needed in which it could be broken down in a very systematic manner, and this one is again used as a raw material back for the synthesis of rhodopsin. Now we are going to go a little further deep into it. So the light strikes the retina. So what are different biochemical changes that happen? So we call it like membrane hyperpolarization, generate a nerve impulse. So what is this membrane hyperpolarization? So remember the surface membrane of a nerve which is generally negatively charged. So that is what we call it like the resting potential. The moment impulse goes across the nerve, okay, the polarization happens, there is a reversal of the charge. For a brief nanosecond, the negative charge becomes the positive charge. This is how an impulse gets along the nerve. So hyperpolarization of the membrane is brought about by a visual cascade involving cyclic GMP, guanosine monophosphate. So this is the second messenger involved in the conversion of polarization status of the membrane, what we call it like hyperpolarization. So you see over there the rhodopsin, when the photon hits up, you have the meta-rhodopsin, the final product, then the GTP gets converted to GDP, there is this transducin which becomes active, and then you have the phosphodiesterase getting converted into inactive phosphodiesterase. The inactive one gets converted into the active form, and the phosphoesterase cleaves the cyclic GMP to give 5' GMP. So here you have the visual cascade involving GMP. So any decrease in cyclic GMP closes the sodium channels in the membranes of the rod cells. So rod cells are, I would say, nerve cells modified, adapted to suit the needs of the visual detecting ability of the eye, especially the retinal layer of the eye. So this result in hyperpolarization, where you have a excitatory response transmitted through the neural network to the visual cortex of the brain. In other words, what happens in the biochemical level would now go further as a signal change in the cellular level involving cyclic GMP, and cyclic GMP is a second messenger system that utilizes the retinol conversion in the wall cycle. So you see over there the sodium channels of the membrane closes. Generally sodium channels open very briefly, allowing a rapid reversal of charge, because you find too much of sodium ions outside the cell and too much of potassium ions inside the cell. So when I mean the cell, I mean the neuronal cells. In this case we are looking at the cells present in the retina. So they are the ones which are responsible for this action, what we call it as the rod cells doing the job of getting activated and transmitting an electrical signal across. Not only that, for color vision, so cyanopsin you have the blue color, porphyropsin you have the red color, iodopsin you have the green color. So all these are basically color sensitive pigments. So as we do light correction, color corrections, when we do editing and do all the photo work, constantly in our eye such kind of color adaptations are being happening. And it is very interesting because except for primates, humans and chimpanzees and very few animals, none have this ability to have or see the world in color. So most of them, they rely on black and white images and interpretation, although it is very accurate and sharp, but the color based interpretation of the environment is ability seen in primates, especially human beings. And you see over there VO retinal, the vitamin A, for all these pigments which play an essential role in giving rise to the shades of colors that we see and appreciate. You see over there, these pigments are basically the protein and the retinal combinations. So protein retinal combination gives rise to all these varieties of pigments, and each of the pigment, depending upon the kind of combination of this vitamin A in them, in this case retinal in them, gives rise to variations in which the information can be conveyed across. So, you see that each pigment being corresponding way of being bleached passes a nerve signal, and the overall blend of the impulse will give the interpretation of image in the brain in alignment with the stored information in different parts of the brain. So, now we are going to go for the second function. So far whatever we have seen all pertains to the function of eye playing a critical role in utilizing vitamin A. Now we are going to look at how vitamin A is going to play an essential role in growth and development. So, you see over there retinoic acid is extremely essential for cellular differentiation, especially in the early stages of embryo development we require sufficient doses of vitamin A. That is one of the reason pregnant women are recommended to take sufficient doses of vitamins, although I have told you earlier it is mandatory for them to take folic acid as a supplement. It is also understood that they need to have sufficient amount of these vitamins in their proper diet. So, you see over there the retinoic acid is essential in the development of epithelial cells. So, in the early stages of embryonic development you require plenty of these epithelial cells which differentiate into other category of cells. So, if there is any impairment in the formation of epithelial cells, and if you could directly associate with deficiency of vitamin A, the effects that would cause would be enormous. It can even lead to abortion of fetus. So, you see there epithelial cell integrity. Now, look here, for epithelial cells to differentiate, epithelial cells to be maintained, you need vitamin A. Lack of vitamin A can lead to dysfunction of epithelia. So, now I want to summarize today's session. So, we have seen in detail starting from the storage of vitamin A, and we proceeded further in looking at the use of vitamin A as an essential vitamin in the way by which vision is realized in the eyes. So, we saw Wahl cycle, we understood the pathway of how vitamin A being systematically converted in forming rhodopsin, the visual purple, and we saw dark adaptation, ability of our eye to really understand the process of conversion of electrical impulse being generated in the eye. The typical process of vision, we owe vitamin A. So, today's session we also saw how systematically it is been converted from the inactive form to the active form, and now it is been constantly being replenished from the liver through blood and finally reaches the eye. So, we focused in detail on the integrity by which vitamin A is essential for dark adaptation. Thank you once again for your attention.